Screening strategies. After the construction of genomic or cDNA libraries, next step is to detect a target clone. Before going to discuss the screening strategies, again I briefly tell you about the definition of genomic library. If you remember, genomic library, it contains recombinant fragments that are ligated into a suitable cloning vector, and they represents the whole genomic makeup of a particular organism. On the other hand, जो cDNA library है, ये messenger RNA की जो population है जो कि extract किए गए हैं from particular tissue, ये उसको represent करती है. अब हम different screening strategies को discuss करेंगे जो कि particular clone को from the thousand of clones इसको detect करने के लिए use की जा सकती हैं. These screening strategies, they may be based on some genetic methods, or screening may be sequence dependent or expression product of the clone it can be detected. As far as sequence dependent screening is concerned, here we can use DNA hybridization techniques, or PCR methods can also be applied for this purpose. If we are going to use PCR method, here we need a set of primers that can detect a particular clone. And here keep in mind that PCR, it is not only used to detect a particular target gene from uncloned or genomic DNA, it can also be used for the screening of a particular clone from genomic or cDNA libraries. On the other hand, if the expression libraries are need to be screened, then the prerequisite may be that the cloned fragment must be expressed so that it may produce functional biological product that may be a protein. Now, protein after expression, it can be detected by using different type of techniques. For example, antibodies that are specific for that expressed protein, it can be used. Antibodies, they are highly specific in their reaction, and they can react with a particular target protein. So antibodies can be used to detect the expression proteins. On the other hand, biological function of the protein, it can be assayed by using a suitable biological system. So we can say that screening strategies, they may be based on genetic methods, or sequence dependent screening may be there, and screening of expressed libraries can be there. And first we can focus on genetic methods. And one of the genetic method, it is selection for the presence of vector. If you remember, while we were discussing different type of cloning vectors, then its detection, it was based on different methods. As far as selection for the presence of vector is concerned, different type of microbiological methods can be used in combination with genetic methods, because when we are going to cultivate microbes that contain our recombinant vector, then it can be detected by using different selective media. If plasmid or cosmid, they are used as a cloning vector, then they contain different type of selectable markers. और ये जो selectable markers हैं, इनमें drug resistance की genes हो सकती हैं, metal resistance की gene हो सकती हैं, or some nutritional markers may be there. If the selection, it is based on drug resistance, so here after introducing the recombinant vector, that may be a plasmid or cosmid, then the host organism can be grown in the presence of selection pressure, like the antibiotics, like chloramphenicol can be used, ampicillin may be there, tetracycline may be there, and then the recombinants that contain our gene of choice, it may be able to grow on antibiotic supplemented medium. So only those microbes can survive that have the resistance against antibiotics. So in this case, antibiotic, it can act as a selectable marker. On the other hand, if we have a look on other cloning vector like the bacteriophage lambda, here the different type of lambda vectors can be used, like replacement vectors are there, insertional vectors are there, and their selection, it may be based on different strategies. For example, in case of replacement vector, where we can clone around 23 kilobase DNA fragment, here the size of the packaged DNA, it will act as a selectable marker. And those other phages that contain only the viral DNA arms, like the right or left arm without the insertion of the foreign DNA, they are too small to be packaged. So the viable phages will not be produced. So here the size of the genomic DNA, it will act as a selectable marker. On the other hand, in case of insertional vector, if you remember, the inactivation of beta-galactosidase gene, that when a foreign DNA is not inserted, this gene of beta-galactosidase, it will keep on expressing. So the plaques that are produced, they will be blue in color, because beta-galactosidase, it will degrade the X-gol chemical that is present in the medium and convert it into a blue color. So blue plaques indicate the ligation of the vector without the insertion of foreign DNA. On the other hand, if foreign DNA is inserted, the expression of beta-galactosidase gene, it is inactivated, and we will get colorless plaques. So in this case, this strategy of genetic methods, it can be used to differentiate between the recombinant vector or the native DNA, or in other cases the parent vector, it can also be differentiated. So this is one of the strategy by which we can use genetic method to detect our recombinant vector.